18, Controlling Bacterial Growth. Problem: how can you determine the effectiveness of an antibiotic? Introduction. Have you ever had an ear infection or strep throat? If you have, you've likely used an antibiotic to treat the infection. Antibiotics work by blocking the growth and reproduction of bacteria. Doctors have to choose the right antibiotic for each infection because not all antibiotics work on all types of bacteria. For example, some bacteria have an outer membrane that surrounds the cell wall. Antibiotics that cannot pass through this membrane have no effect on those species of bacteria. Bacteria that are not affected by an antibiotic are said to be resistant to that antibiotic. Over time, there has been an increase in antibiotic resistance. Bacteria that used to be killed by certain antibiotics are no longer responding to them. Some bacteria, such as MRSA, have built up many resistances. Scientists are trying to produce new drugs to kill these so-called superbugs. In this lab, you will test the effectiveness of antibiotics against two kinds of harmless bacteria. One of the bacteria has an outer membrane and the other does not. Materials: 8 nutrient agar plates, a marking pen or labels, 2 bacteria cultures, 1 Escherichia coli or E. coli, which we will denote as EC, and 1 Staphylococcus epidermis, which we will denote as SE. 32 sterile glass beads, disposal solution, 2 sterile micropipettes, 8 forceps. These have been sterilized using heat. 8 paper discs, 2 tetracycline, which we will denote TCN, 2 clindamycin, which we will denote CLI, 2 cephalexin, CEF, and distilled water H2O, masking tape, metric ruler, and incubator. Currently our temperature is 29 degrees Celsius, but it will warm up. And here's a look at the inside of our incubator. We have a temperature probe in there, and you can see that depression on the left. That's for incubating samples in taller bottles, like flasks. We'll be putting our agar plates right on the higher elevation. For safety, don't forget your goggles and gloves. We will also need to pay close attention to sterile technique and hand washing, and make a note of any allergies to tetracycline, clindamycin, or cephalexin. Procedure. Label the agar plates. We're going to label the agar plates accordingly. Each plate will have a number as well as a notation for which bacteria has been cultured and which antibiotic disc has been placed. So plates 1 through 4 will be for E. coli, plates 5 through 7 for S. epidermis. TCN, CEF, CLI, and H2O will denote the contents of the paper discs placed on the agar plates. Our top row of plates will be for our E. coli cultures, and our bottom row will be for our S. epidermis cultures. Add culture to agar plates. Put on your goggles and gloves using a sterile pipette. Add 3 to 4 drops of the E. coli culture to plates 1, 2, 3, and 4, removing and replacing each lid quickly. And then we will repeat this process using a new sterile pipette and adding 3 to 4 drops of S. epidermis culture to plates 5, 6, 7, and 8. We will quickly remove and replace each lid. forget to put on your gloves and goggles first. Now we will thoroughly inoculate our agar plates. We're going to work with one plate at a time, remove the lid, add 4 sterile glass beads to the plate, replace the lid quickly, and then repeat this for each plate. We're going to then tilt and swirl all the plates to spread the culture. And then, one plate at a time, we're going to remove the lid quickly, pour the beads into this disposal solution, and then replace the lid quickly. These glass beads have been heat sterilized. Now we're ready to add our antibiotic discs to the plates. We're going to work one plate at a time. Quickly removing the lid from plate 1, using sterile forceps, we're going to press 1 tetracycline disc firmly onto the agar off-center in plate 1. We're going to replace the lid, and then we're going to repeat this process for plate 5. For plate 2, we're going to use the same process and place a disc of cephalaxin onto the agar for plate 2, and then also plate 6. We're going to use the same process for plate 3 with clindamycin, and as well for plate 7. And then we're going to place our distilled water discs in plates 4 and 8, and we're going to use a new pair of heat sterilized forceps every time. It's now time to incubate. We're going to tape down each plate's lid, invert the plates, and incubate them at 37 degrees overnight. This is our brand new homemade incubator, which does a pretty good job of maintaining a temperature right around 36, 37 degrees Celsius. And our incubator has a heating pad in the lid, aluminum tape all over the inside, as well as aluminum foil and insulating foam. Here's what everything looked like before we closed up the lid. These straps help hold it down tight and keep the heat inside. Safety precautions. Remove your gloves properly inside out and throw them away. And then don't forget to wash your hands. Use soap. Observations. Observation and data collection. So we're going to examine each plate and we're going to measure the diameter of any clear zone around each antibiotic disc. Before we do that though, I want to give you a little bit of data background. So we have gram-positive and gram-negative bacteria. The cell walls of gram-positive bacteria are made up of a thick, mesh-like layer of peptidoglycan. Gram-negative bacteria have only a thin layer of peptidoglycan in their cell walls, but they also have an outer membrane containing lipopolysaccharides. So having a look here at the antibiotics we've chosen, the classes to which they belong, and the types of bacteria against which they are effective, we can come up with some hypotheses. So tetracycline is a general class, actually, and they are broad-spectrum antibiotics, and they work against both gram-negative and gram-positive bacteria, as well as some other atypical organisms. So E. coli is gram-negative, so we should hypothesize that tetracycline would be effective. And S. epidermis is gram-positive, and we could hypothesize that, again, tetracycline would be effective because it is broad spectrum. Clindamycin is a lincomycin antibiotic, and this is a narrow spectrum antibiotic that is effective against gram-positive and any bacteria that does not have a cell wall, and that includes species of strep and staph. We were using one of those species today. It's also used for people who can't take penicillins. So again, E. coli is gram-negative, and we should expect in this case that the E. coli would be resistant to the clindamycin because it is gram-negative, and clindamycin is effective against gram-positive bacteria. The S. epidermis, however, is gram-positive, so we should anticipate it would be effective. Lastly, cephalexin is a cephalosporin. First-generation cephalosporins are very effective against gram-positive bacteria, but only somewhat effective against gram-negative bacteria. Cephalexin itself is a first-generation cephalosporin. So with E. coli being gram-negative, we can say that they are somewhat effective. So I'm going to put a little question mark for our hypothesis there. We're not quite sure. And for gram-positive bacteria like S. epidermis, we should expect that the cephalexin would be effective. Well, here we are the next day, and it looks like we've maintained a nice 36 degrees Celsius. So we're going to put on gloves, open up our box, and see what we've got. Well, here they are, and you can see we did fix those labels. So we now know what we're dealing with here. So I'm going to take for your first one here, plate number 1, and this is your plate with your E. coli and your tetracycline. So what we're looking for is a clear zone around the antibiotic. So one at a time, I'm going to lift up your plates so that you can see them with a little backlighting, because I found that's the best way to get a clear image. So our first one here is our E. coli exposed to our tetracycline. Now you should be able to see your bacteria growing pretty clearly, all of that. If we get it nicely backlit at just the right angle here, it should pop out for you. Do you see that clearing around your antibiotic disc? That means that your E. coli is susceptible to being destroyed by your tetracycline. You can see that nice clearing around your disc. That's a beautiful sample. All right, so I'm gonna put this one down, and we're gonna go next with number 2. This is E. coli exposed to clindamycin. You can see we fixed the labeling issue we had before because I had all my little bottles in the wrong place when I was filming. All right, here we go again, clindamycin. We're looking for a clearing. Do you see a clearing? I see a clearing right there around that antibiotic disc. Maybe not quite as obvious as the last one, but it's there. Okay, and our next one will be our E. coli and our cephalexin. This one is beautifully done. Look at that. Perfect. That is a nice clearing. So far, all 3 of our antibiotics are going to cause issues for E. coli. and could be used to fight an E. coli infection. Let's have a look at our distilled water. There we go. Do you see a clearing around the disc? I don't. So distilled water had no effect on E. coli growth. All right, let's have a look next at our Staphylococcus epidermis. So here's our staph exposed to tetracycline. So that for you. Oops. So we're looking for a clearing. Again, do we have a clearing? I can get you a good angle, sorry. That looks like a good potential clearing. Maybe not as clear as the others, but I would say that's a yes. Looks to me like a clearing. And let's have a look at our staph exposed to the clindamycin. I'm going to get you the right angle here. What do you see here? I see a huge clearing, really big. Goes almost to the center of the plate. So this is a very effective antibiotic for staph infection. And then let's try our staph exposed to our cephalexin. Again, what are we looking for? Clearing. Do we see one? I see another really big one. You see it? You can see right there where the margin, you can really clearly see this margin right here. And then lastly, let's have a look at our staph with our distilled water. Correctly. So this one was a little tricky, but there isn't a clearing. You can actually see some of the lines pretty much run through it, or darn close to through it. It looks like this disc was actually still a little bit wet and it just diluted some of the sample. But if you get, I can get it close enough to the camera. Yeah, you can see the lines go through it. There's not really a clearing there. So I'm gonna get some measurements for you. So here's our data table. I took measurements around 12 hours, and I'm going to take them again at 24. So we're seeing everything that we would expect, except that we should not be seeing any clearing on the plate with E. coli and clindamycin, and we did see some clearing. And then also, it looks like we are seeing clearing on plate 3 with the cephalexin and E. coli, and we weren't sure what to expect there. Analyze and conclude. You're going to check your Lab 18 assignment in Teacherease, and you're going to download and print Lab 18 Analyze and Conclude answer sheet. Complete the answer sheet and turn it in class on 111. So what's the purpose of the disc soaked in distilled water? Is there any evidence of antibiotic-resistant bacteria on your plates? Explain which antibiotic or antibiotics are effective against each type of bacteria. Some antibiotics are less effective against a specific type of bacteria after prolonged and repeated use. What do you think is responsible for this pattern? Hint, think about the principle of natural selection. Suppose you are prescribed an antibiotic to treat a bacterial infection. Your doctor tells you to take the antibiotic for 10 days, even if you feel better before then. Why should you continue taking the antibiotic for the full 10 days? And that's the end, folks. See you back in class.